Hello everyone and welcome to today's webinar. I am Sunit, Senior General Manager for Biologics Services at Syngene. It's a pleasure to have you all with us. Syngene is India's largest CDMO and it was started by Kiran Mazumdar-Shaw around 30 years ago. And we proudly say that we work with 15 out of top 20 global pharma companies for their project. Today, we are diving into how you can elevate your protein production with SynWeave, our innovative platform that enhances manufacturability and significantly boosts productivity. I'm excited to share insights and solutions to help you take your biologics manufacturing to the next level. Please note, we'll have a live Q&A chat session during the webinar. Feel free to drop your questions in the chat box and we'll address them throughout the session via chat. Any unanswered questions will be followed via email, or you can write to us directly at bdc@syngeneintl.com. So let's get started. Let me introduce SynWeave, the platform which we recently launched at Syngene. This platform significantly elevates protein production, keeping the quality parameter in mind. Our goal with SynWeave is to address the key challenges in clone generation and also enhance the efficiency of the production process. In my follow-up slides, I'll speak about the salient features highlighting this platform, which will significantly overcome the bottlenecks in cell line development. But before we move further, I think we should discuss about the CHO cell line, the history about it. I think we all are aware that Chinese hamster ovary or CHO cells are a source of multi-ton of quantities of biopharmaceuticals. CHO cells have been known for decades to be highly robust and flexible, one of the main reasons for their popularity in manufacturing. CHO cells have been used in culture for more than 50 years, and various cell lineages are available and have been used in manufacturing. These cells initially were established in the laboratory of Dr. Theodore T. Puck in 1957, and this outbred hamster was provided by Dr. George Yerganian of the Boston Children's Cancer Research Foundation. CHO cells comprise a variety of lineages, such as DXP11, CHO-K1, DG44, CHO-S, etc., that share a common ancestor. CHO-K1 was derived from a subclone of the original cell line. In 1980, the DXP11 was generated through chemical mutagenesis of CHO-K1. In 1983, Ullab deleted both DHFR alleles by mutagenesis of a different CHO cell line starting population, thus generating CHO-DG44 lineages. In 1991, the suspension cell line CHO-S was generated from another CHO cell starting population. CHO-ZN, that has been developed by Mark Millipore, enables rapid development of recombinant cell lines by strong metabolic selection. Nowadays, all these cell lines are widely used for the production of biopharmaceuticals. At the same time, we all are aware that CHO cells as mammalian cell line expression system offer several advantages. The most important being their ability to produce proteins with complex glycosylations, post-translation modifications that are similar to those produced in humans. Therefore, this CHO cell line has been used extensively for therapeutic protein production. Let me take you to the traditional cell line development workflow now, which generally starts with the transfection of gene of interest in CHO cell line of choice, followed by selection, limiting dilution, and cell banking. The major bottleneck of traditional CLD workflow is random integration. We have no control where a gene of interest is integrating, thereby need to screen many clones to get the best producer. At the same time, it's also not possible to screen quality parameter of all the clones at all the times. Finding the right clone with the high titer and desired quality is like finding a needle haystack, and it involves considerable times. The traditional approach of cell line development has two major shortcomings: the low efficiency of integration and the homogeneous population with respect to gene expression at the integration sites. The heterogeneity in gene expression arises primarily due to position effect, which is shown depicted here, the major challenges what we generally observe in traditional CHO cell line development. Let me now briefly describe. As we all are aware from our basic biology that with respect to genomic organization, we have euchromatin and heterochromatin. Heterochromatin is highly condensed, gene poor, and transcriptionally silent, whereas euchromatin is less condensed, gene rich, and more easily transcribed. When a transgene integrates into a region of euchromatin, it is more likely to experience favorable and stable gene expression. But if the transgene integrates into heterochromatin, it is more likely to experience gene silencing or reduced expression. So expression of transgene is dependent of position of genomic locus where it is integrating. It is known as position effect. The problem of position effect occurring as a result of random integration has been addressed by several research groups over past decade. To address the issue, genetically engineered vectors have been produced, which include components such as matrix attachment regions or MARS, scaffold attachment regions or SARS, stabilizing anti-repressor element stairs, and ubiquitous chromatin opening element UCOE. These components can modify the chromatin environment to minimize gene silencing and enhance higher, more stable, and biologically active expression of recombinant molecules. But in spite of these modifications of having SARMAR and UCOE elements in our expression vector, the screening of clones to find the good producer and stable clone is highly labor intensive. Much efforts have been put also in identifying transcriptional hotspot in host cell genome in recent years. The state-of-the-art technique is to integrate not only the individual transgene but also complex cassettes called landing pads to generate master host cell lines. Landing pads contain selection markers and recognition sites for recombinance or integrases, which allows the precise insertion of any kind into the established sites. There are numerous systems have been effectively used for mediating expression cassette exchange by site-specific recombination in mammalian cells, such as KLOX, FLP, FRT, and BXB1 recombinases. In addition, targeted genome manipulation techniques enables the knock-in of recombinant protein coding genes into well-defined and transcriptionally active genomic sites. Targeted genetic manipulation of cells has been made possible by the enhanced emergence of target-specific genome editing tools such as zinc finger nuclease, which is known as ZFNs, transcription activator like effector nuclease or TALENs, cluster regularly interspaced short palindromic repeats, which is known as CRISPR, CRISPR-associated protein 9 or Cas9, RNA-guided nucleases. Among these, CRISPR-Cas9 system became the most popular for its ease of use and its highly editing efficiency. Though we have been some success with targeted integration, further work at the chromosomal level will facilitate the identification of potential target size as well as a better characterization of the correct targeted integration strategy. There is another type of integration which is known as semi-targeted integration, is carried out with the help of transposon system. The transposon is a mobile DNA element capable of transposing within genomes. Transposon system or jumping gene, it was earlier discovered by Barbara McClintock in maize. Transposon system has been recently used as part of the mammalian cell line development, and several publications has shown its effectiveness in generating cell lines. As told earlier, the main advantage in this technology is that it involves a semi-targeted integration rather than the random traditional integration, thereby mitigating the challenges of position effect and also improving the integration efficiency successfully. This slide explains why transposase-mediated transfection is so efficient. When you add the engineered transposase enzyme, the entirety of the DNA construct is integrated into the human genome with very high efficiency at several locations. This so-called translocation occurred through a cut and paste mechanisms. A random integration, as we have discussed earlier, is highly prone to the genetic scrambling as diagrammed here. Because it is so random, integration of a full construct turns out to be a rare event. Even though we can screen and isolate a clone that has the full construct, because it is so inefficient, we have to screen quite a lot to find one of the functional clone. With the transposon system, not only most cells are efficiently transfected, they always get the fully functional construct. As transgene is integrated at multiple locations, this also increases the copy number and integration efficiency. So, as part of SynWeave platform available at Syngene, we have different ways of enhancement of titer of therapeutic protein. We start with cell line development, where we have transposon-based technology, which gives you titer 3 to 4 grams per liter with the monoclones generated. This transposon technology has been in-licensed from an European-based CRO. In addition to transposon technology, we also use our upstream process development platform, thus further enhancing the titer. We have achieved between 1.5 to 2-fold increase in titer using our platform upstream process. So as part of titer enhancement platform, what we offer? We offer transposon-based cell line generation using proprietary CHO cell line, resulting in decreased timeline, developing high-yielding homogeneous clone. Secondly, the platform approach for enhancing titer of cell line with minimum experiments using GOE and AMBER 250 bioreactor. This technology meant to produce maps with high titer and ensures consistent product quality in first time right and quick approach. Now let us go through that our CLD workflow. The genes for expressions are codon optimized for high expression. After the pool selection stage, single cell cloning is performed, and as a part of regulatory requirement, monoclonality data is generated. The single cell cloning is done by cytina, which drops single cell into the 96-well plate. Images are then captured during this event. Further, images are captured by Solentim cell metric on successive days, like day 0, 1, 2, 3, and so on, to show the doubling during the growth. So this gives the monoclonality proof that cell line has arisen from a single clone. For the FC containing protein like IgGs or bispecific, octet HTX system is used for titer estimation, which are high throughput, and the titers are estimated at each stage of the 96-well plate, followed by 24 and 6-well plate. And the clones which are showing high titer are then moved to PQ analysis, we use Amber-based mini bioreactor system to carry out ranking of clones. Top clones are selected not only based on the titer, but also on the quality parameter, including charge, monomer, glycan, and potency. The selected clones are banked and stored in liquid nitrogen canister for further usage. To summarize, the platform, the cell line development stage, we are selecting our top clones both by titer and quality parameter. What we have observed in our SynWeave platform that 70% of the clones we screen having high titer. So in this platform, actually a lot of clones need not be screened to get good producers. This saves a lot of time and the cost of raw materials, which can actually make the cell line development cost very reasonable. What you see, this graph on the left actually shows the distribution of the titer across all the clones analyzed. We can see that 70% of the clones that we have screened almost have a consistent titer of above 2 grams per liter, and thus confirming that through this technology, titer of the clones are fairly uniform, that we don't need to screen a lot of clones to get a high producer. On the other hand, you would appreciate that using our SynWeave platform, we have obtained between 3 to 5 gram per liter titer for a IgG1 molecule without any optimization, as shown on the right, and QP was also on the higher side. In the context of biopharmaceutical production, stability is defined as the reproducible protein yield and quality from a given cell line over extended period of times, from thawing of the cell line from a cell bank until a given time point that is considered the longest period allowed for production. We generally carry out stability of the research cell bank up to 60 generations. What we have observed that clones generated using transposon technology, the product quality is almost consistent across the generations, and also there is no drop in titer. Generally, clones are considered to be stable within 30% variability in the titer across generations. But here what we see, that drop in titer across 60 generations is very less, and the product quality across different generations also is highly conserved, that we don't see surprises once we go to the late generation that there is a difference in charge species or a glycan profile. I think all this is enabled because of the technology that is used, that we get good clones which are very stable and have consistent product quality across generations. As a part of upstream process development, we use Amber 250 system, that is a miniature bioreactor which is used for screening, so that the best producer are screened at the real manufacturing control environment. As part of the upstream process development, we do have all offline parameters like glucose, lactate, ammonia, glutamine, glutamate, etc. Once we have identified the best top producing clone, we move into process development. Again, it is done statistically using a DOE-based approach here. Generally, we do a parametric interaction and we carry out media feed screening, agitation, temperature downshift, as well as feeding regions and initial seeding density. During this process, we also establish a design space for the manufacturability. Once the design space is established, we move into a 10-liter scale to check the design space whether it is scalable from 250 ml to the 10-liter scale. This will be called as a process assessment at 10 liter. So this is how we established a robust manufacturing process for any type of molecule which we are developing end-to-end as part of our SynWeave platform. At the end of upstream process development, we would have developed manufacturing process which is highly robust and scalable. The media feed selection significantly impact product expression and quality. In our AMBER 250 system, we perform clone screening using the most suitable media and feed for the target molecule. By integrating media and feed screening into our top clone selection, we identify the optimal clone before process development begins. To ensure the desired product profile, we assess the need of additional vitamins, amino acids, cofactor during the process development. Parametric studies along with optimized media feeds and supplements help establish optimal process conditions. Finally, as I told earlier, a design space is used to lock in the process at 10-liter scale. This is a case study of upstream process optimization of in-house developed clone. Using a design of experiment DOE approach, platform interaction in near manufacturing setup, we screened 24 bioreactor conditions to identify the optimal parameter for clone growth, medium compositions, and process conditions. We selected 10 conditions for further analysis based on titer, specific productivity, and purification results. Thereafter, the top condition was scaled up to 10-liter bioreactor. With minor process adjustment, we achieved a titer of 7.2 gram per liter, representing a 4-fold increase in titer compared to previous processes. This platform approach significantly improves efficiency and reduces cost for the biopharma company by producing high-quality mAb clone with industry-leading yields. Let me take you all through another case study with a client-provided cell line. Client had a process of 22 days and the titer of 0.8 gram per liter here only. So our task was to reduce the process time and also at the same time increasing the titer. We were able to do a similar kind of media feed screening and parametric interactions in our AMBER 250 as part of our SynWeave platform, what I described earlier. At the end of our DOE study, we could achieve the titer of 4.2 gram per liter and also reduce the timeline from 22 days to 17. All the parameters were identified for this same space without compromising the quality. This process later was scaled up to 10 liter and then even up to 500 liter at manufacturing. We also have got the similar titer of 5 grams per liter even at 500 liter scale and significantly reducing the timeline for the GMP manufacturing from 22 days to 70 days process. This is another success story of our SynWeave platform. To summarize, SynWeave was designed to significantly improve the biopharma manufacturing process through 3 core advancements. Number 1, producing high titer clones. SynWeave is engineered to optimize clone yields, ensuring that we achieve maximum concentration and effectiveness. This high titer capability is crucial for delivering superior results and meeting the demanding need of biopharma production. Secondly, shortening timeline. With SynWeave platform, we have streamlined development and production timeline. By accelerating these processes, we can bring our solutions to market more quickly, keeping pace with the rapid evolution of biopharma industry. And thirdly, creating clones with high manufacturability. Our platform enhances manufacturability, reducing the risk associated with scale-up production. This means that our clones are not only effective, but also easier to produce at a larger scale, minimizing potential hurdles in the manufacturing process. The development of SynWeave combines transposon-based gene expression technology with the extensive process development experience of a Syngene scientific team. This integration of advanced technology and deep scientific expertise has enabled us to create a platform that stands at the forefront of biopharma manufacturing innovations. But we are not ending here with Titer enhancement. Currently, we are also working on N-1 perfusion with high cell density fermentation batch to increase the titer further. We have already generated data that we can increase the titer by around twofold by N-1 perfusion. Now let me talk briefly about Syngene. We are the largest biologics CDMO in India, and we are one-stop solution for all your needs. We have both mammalian and microbial capabilities. In microbial, we work with E. coli, Pichia, algae, and fungal cell line. In mammalian, as I have told you, we have tremendous experience with Chinese hamster ovary cell line. We work on different kind of modalities of the molecule, including MAP, ADC, bispecific recombinant proteins, microbiome, peptide growth factors, PD, and mRNA. I would also like to highlight that at Syngene, we have a GLP compliant viral testing facility where we perform clearance and characterization studies. We have a state-of-the-art analytical laboratories which supports throughout our process development and also carry out standalone analytical characterization studies. We have been trusted partner for customer, providing solutions to several challenging projects at Syngene. We have also recently developed other platform where we can able to provide GMP grade DP to initiate clinical trial in 10 months. What I mean to say that from the gene sequence to GMP DP for IgG1 class of molecules, will be able to deliver the product in 10 months to initiate our clinical trial. As I've shown you earlier, we have fed batch productivity of IgG1 class of molecule between 3 to 5 gram per liter. Using our fed batch process, we are able to increase by a couple of fold and intensified fed batch process with N-1 perfusion. We have started integrating that titer enhancement into our titer enhancement platform. Capacity wise, we have more than 20 KL bioreactor already installed. These are single use bioreactor. We have a GLP certified viral testing facility, and also quality control lab is a new state of the art which has been already established at Syngene. We have more than 20 projects have been delivered from development to R&D. We have carried out several early stage and late stage projects, analytical services, and even several end-to-end projects we have delivered successfully. So thank you all for joining today's webinar on SynWeave. We hope you found the insights valuable and that our innovative platform can help you take your biologics manufacturing to the next level. If we missed answering any of your questions, rest assured, we'll follow up via email. As I told you earlier, you can also reach out to us directly at bdc@syngeneintl.com for more details. We look forward to collaborating with you and supporting your biological journey. Thank you again and have a great day.